So what if we could peek inside the brain? Not just for a moment, but, for longer periods.
Right, actually capturing the activity of individual neurons.
Exactly. And without needing really invasive surgery.
Hmm.
Imagine the kind of understanding we could unlock.
It sounds almost futuristic, doesn't it? That level of detail.
It really does.
Yeah.
But today, we're diving into research that's bringing it, well, remarkably closer to reality.
Yeah.
Welcome back to the Deep Dive. Today we're unpacking some pretty groundbreaking science, turning complex research into hopefully compelling insights for you.
Mm-hmm.
Our mission today, to explore this incredible leap forward in neuroscience. We're talking about a super miniaturized wireless image sensor.
Tiny. Designed specifically for brain fluorescence imaging.
Yeah, a device so small it really pushes the boundaries of what we thought was possible for, observing the brain's most fundamental units.
It's a truly pivotal development, I think. And our source for this deep dive is an academic paper. It's titled A Miniature Wireless Silicon-on-Insulator Image Sensor for Brain Fluorescence Imaging by Zhang and colleagues.
Okay.
And this paper, it really lays out the conceptual design, the initial prototypes, and, well, the fascinating journey of making this tech a reality.
And the why it matters here is huge.
Oh, absolutely immense, because understanding the brain's complex mechanisms, it really requires observing neurons in their natural active state.
Which our current tools really struggle with.
They really do. It's been a long-standing problem.
Exactly. So for you, our listener, our goal today is to really get under the hood of this microscopic device.
Here.
Understand how it works, why it's such a game changer for neuroscience.
And what it could mean down the line.
Yeah, what it could mean for understanding the brain's fundamental processes. Because let's be honest, the brain is still, well, it's a vast, largely uncharted territory.
Definitely.
And being able to observe its activity in real time at a cellular level, that feels absolutely crucial for navigating it.
Indeed. And the paper highlights this fundamental challenge neuroscience has always faced, observing neural activity in freely behaving animals.
Right, not just in a dish or under anesthesia.
Precisely. It's not just a technical hurdle, it's critical for truly understanding complex neural mechanisms in their natural context.
Like watching a bird sing while it's actually flying, not just looking at its vocal cords in a lab.
Exactly that kind of analogy. If you want to grasp how brains truly work, we need to observe them undisturbed in a natural state.
But our tools have always gotten in the way, somehow.
Yeah, they've imposed significant limitations for a long time, preventing those precise, long-term, unencumbered observations without, causing damage or interference.
Mhm.
And what's fascinating here is how this new device tries to overcome those long-standing hurdles, pushing past limitations that have frustrated researchers for decades.
Bringing us closer to maybe a transparent interface with the brain.
That's a great way to put it, yeah. Closer to that ideal.
And those limitations, those persistent problems, that's really where we need to start, isn't it?
Absolutely. To fully appreciate this new sensor, we first need to understand just how difficult brain imaging has been and, well, still is.
Okay, so let's talk about the current landscape. Why is it so hard to get that clear, unencumbered view of neurons doing their thing?
Right. It's a complex picture with lots of trade-offs.
So let's start with a common one. Multi-electrode recording. Sounds like it captures electrical signals.
It does. It's excellent for capturing population-level electrical signals, understanding the overall rhythm, say, of brain regions.
Okay, the big picture.
The big picture, exactly. But when it comes to specific individual neural signals, well, what does it really tell us? That's the question.
What's the main issue there?
Well, the paper points out it lacks the selectivity. You can't unambiguously track well-defined cell populations, or certainly not single cells, over long periods. Think of it like listening to a whole orchestra, or maybe just the string section. You get the overall sound, the dynamics, but pinpointing one specific violin and tracking just its part consistently over days or weeks, that's incredibly difficult.
Right. You get the broad strokes, not the fine detail of each player.
Precisely. Lots of information, but not specific enough for certain questions.
Now, that orchestra analogy really helps visualize it. So that gives us broad strokes. How do researchers even start identifying specific pathways if they can't isolate cells? Has anyone figured out how to make specific neurons, like, light up?
Ah, that's a great question. And it leads us right to genetically encoded calcium indicators, or GECIs.
GCIs. Okay.
These are really key for getting single-cell level genetically selective measurements.
How do they work?
Researchers can basically modify specific types of neurons, say only those involved in memory or a particular neurotransmitter system, so that when those specific neurons become active, calcium rushes in, and these GECI proteins inside them, they light up, they fluoresce.
Wow. Like putting tiny bioluminescent tags on just the cells you care about.
Exactly, like tagging just the violins you want to track in that orchestra. It gives you amazing specificity. You know which cells are active.
But there's always a but, isn't there?
There often is. So while GCIs offer that specificity, the problem then shifts to the imaging tools you use with them.
Ah, how you actually see the light.
Right. And those tools often suffer from this major trade-off. You either get really high resolution, a detailed picture, but you might have to cause significant damage to the brain tissue to get that view, or you choose a less invasive approach, but you sacrifice the resolution you need to clearly see individual cells firing. It's this constant balancing act.
And that balancing act seems to be the core problem. So another tool mentioned is fiber photometry. That sounds a bit less invasive.
It is, yeah. Fiber photometry is definitely a step towards less invasiveness.
What are its pluses and minuses?
Well, the benefit is minimizing tissue damage. It uses a very thin optical fiber inserted into the brain to deliver the excitation light and collect the fluorescent signal.
Okay. Thinner is better.
Thinner is better, and it also allows imaging from deeper brain regions with less disruption than some other methods. So, good for depth, less damage.
But what's the compromise? There's always one.
The compromise, as the paper notes, is resolution. It's limited. You might get a signal from a population of neurons with GECIs in a region showing overall activity.
But not single cells.
Exactly. You can't distinguish the individual firing patterns within that group. It's like hearing the general hum of the violin section again, but not the distinct notes from each instrument.
So you gain depth and reduce damage, but lose that granular detail.
Precisely.
Okay. And then there's miniature single-photon endoscopy. That sounds like it might give a broader view, maybe record many cells at once.
It can, yes. That's its advantage, recording simultaneous activity across many cells, which is great for understanding networks.
Sounds promising. So what's the catch, especially for deeper brain regions?
The catch is invasiveness. Targeting deeper regions often requires highly invasive procedures.
How invasive?
We're talking about inserting relatively large lenses, sometimes Girard lenses, or even aspiration, which means physically removing brain tissue to clear a path for the endoscope.
Wow, actually removing tissue.
Yeah, and the damage caused by these procedures, you just can't ignore it. It can affect the very neural activity you're trying to observe.
Or at least make stable, long-term recordings really hard.
Extremely challenging. It's like, tearing down part of the concert hall wall to get a camera right up close. Great view, maybe, but the disruption is massive.
Okay, so invasiveness, lack of specificity, low resolution. These trade-offs are persistent. It makes total sense why researchers have been pushing hard to shrink the cameras.
Absolutely. They've been relentless, trying to miniaturize for direct brain insertion.
So what were some of those key attempts? Where did they push the boundaries, and where did they eventually kind of hit a wall?
Well, there have been some really clever approaches. Back in 2005, Okada and colleagues first proposed this idea of implantable microimaging sensors for GECIs. That was a big conceptual leap.
Moving from external observation to internal sensing.
Right. Then, Adams et al. In 2008 demonstrated a flat, lensless fluorescence camera. They put a patterned mask very close to the image sensor.
Lensless. Interesting. How thick was that?
The whole system was about 1 millimeter thick, a significant reduction at the time, definitely flatter than bulky endoscopes.
1 millimeter. Still sounds pretty chunky for embedding in brain tissue, though, especially when neurons are measured in micrometers.
It is, absolutely. Scale is everything here. So following that, Shin et al. In 2013 had a different approach. They coupled a miniature mask to a fiber bundle.
Like an endoscope, but maybe smaller?
Kind of, yeah. No extra microlenses needed. It could target deep regions. But the fiber bundle itself was still 400 micrometers in diameter.
Okay, smaller than a millimeter, but 400 microns is still quite large relative to neurons.
It is. Less damage than bigger systems, sure, but still significant disruption for truly minimally invasive long-term studies.
And I recall reading about Choi et al.'s work. That sounded like it got even smaller, really pushing the limits of what was possible then.
Yes, Choi et al. in 2017 really pushed the envelope. They used thin shanks with single-photon avalanche photodiodes, CISPAs. These could record fluorescence along the insertion path, and through some clever post-processing, removing extra silicon substrate, they got the shank thickness down to around 40 micrometers.
Oh, 40 microns. That's less than a human hair.
It is genuinely small, a huge achievement in miniaturization. But this is where they hit that critical, fundamental limit.
A limit with the standard silicon manufacturing.
Exactly. With conventional bulk silicon processes, if they tried to thin that bulk silicon beyond about 40 micrometers,
To break the sensor.
it would physically damage the photodiode components embedded within that silicon layer. You just couldn't make it any thinner without destroying the core sensing element.
Because the light sensing parts were physically inside the silicon chunk they were trying to thin.
Precisely. That 40 micrometer barrier was the physical wall for that technology.
And that is exactly the barrier this new paper, Zhang et al., aimed to break through.
Yes, that's the setup.
Okay, so this is where it gets really interesting. They needed a completely new approach to get past that 40-micron wall.
Yeah.
What was the aha moment? What fundamental shift let them bypass those limits?
The real breakthrough, the aha moment, was realizing they needed a different foundational material process entirely.
Not just tweaking the old way.
No, a fundamental shift. The inspiration, the authors note, came from earlier work on photodiodes designed using silicon on insulator, or SOI, processes.
SOI. Okay, tell me about that. Why is that the key?
It's absolutely key because conventional photodiodes in bulk silicon hit that thinning limit, but photodiodes in SOI, specifically these lateral pine photodiodes.
Lateral, meaning sideways.
Sort of, yeah. Their active sensing area is built more laterally along the surface, not deep into the bulk silicon.
Oh.
And that inherent structure means they are just physically much thinner to begin with. It allows the researchers to then thin the substrate much further.
Pushing past the 40-micron barrier.
Exactly. Enabling device thicknesses below 40 micrometers. The goal is around 25 micrometers.
Less than a human hair diameter, aiming for like a third of that.
Right. And this isn't just a number, it's a massive reduction in invasiveness. It's about minimizing that foreign presence in the brain.
Getting closer to that transparent interface idea.
Exactly.
So SOI makes it incredibly thin. That's huge. But why else is it optimal for this kind of wireless implantable device? Are there other advantages?
Oh yes, several profound advantages. First, as we said, the thinness. Lateral PN diodes let you thin the device way down while still capturing light efficiently from the back. Less disruption, more stable recordings.
Less disruption, paramount goal. Got it. What else?
Another absolutely critical advantage, the SOI structure includes something called a buried oxide layer.
Okay.
And this layer is transparent to light.
Transparent. That sounds important.
Hugely significant because it allows the image sensor, once thinned, to operate using backside illumination.
Light comes in from the back of the chip.
Exactly, not the front. And the implication is massive. It gives designers freedom to put stuff on the top metal layers without worrying about blocking the light.
Like the wireless components.
Precisely. Things like the RF inductors and capacitors, which are necessary for wireless power and data, but are usually quite large. They can be placed directly on top of the photodiodes. This saves crucial area, keeping the device incredibly small.
If you had to light it from the front, those components would block the view.
Completely. It wouldn't work effectively. So backside illumination enabled by SOI is essential.
That is incredibly clever engineering. Using the back for sensing, the front for wireless stuff, without interference. What about power efficiency? That must be huge for a wireless implant.
Huge. And again, SOI shines here. These processes inherently have higher power transmission efficiency compared to standard bulk CMOS processes.
So just fundamentally better for wireless power.
Yes. It makes SOI an optimal choice for low-power wireless applications, which is exactly what this sensor is. Every bit of efficiency counts when you're transmitting tiny amounts of power wirelessly and trying to avoid heat buildup.
Okay, so let's visualize this thing. The paper has that conceptual drawing, figure 1. Paint a picture for us. How small is it really, and what are the main parts?
It's astonishingly small. We're talking about a chip that's just 300 micrometers by 300 micrometers.
Like a few grains of salt?
About that size, yeah. And the thickness? Just 25 micrometers.
25. Less than a hair's width.
Much less. Average human hair is maybe 70 micrometers, so incredibly thin. And it's designed so you could inject multiple units directly into the brain.
Inject them. With what?
Using a very small needle. The paper says less than 24 gauge.
Wow. A 24-gauge needle is tiny, like for fine injections.
Exactly. Finer than a standard blood draw needle.
It's hard to even grasp something that complex being that small. What about the components on it?
So on top, you have the RF coil for power and data, plus capacitors, right on top of the pixel array.
Thanks to that backside illumination trick.
Correct. Underneath is the pixel array itself, those lateral PAN photodiodes looking out through the backside into the brain tissue.
And how does it get power? Wirelessly.
Yes, wirelessly. An external power transmission coil, placed on the scalp, inductively delivers power down to the chip.
Which brings up that absolutely fundamental question.
How do you power something so tiny, wirelessly, reliably, and safely inside the brain?
Exactly. That wireless power puzzle is huge.
It is, and that's the next critical piece we need to unpack.
Right. Powering this miniature marvel seems like a monumental engineering challenge. The paper mentions LC resonator design trade-offs. What does that mean for getting energy to something the size of salt grains?
It's a huge challenge. It's all about balancing efficiency and physical size. Generally, power transmission efficiency goes down as the frequency of the LC resonator goes up.
Okay.
But that frequency is inversely proportional to the physical size of the inductor, L, and capacitor, C.
So small components mean high frequency.
Usually, yes. But high frequency often means lower power efficiency for the transfer.
Ah, the trade-off.
Exactly. You need tiny components for the implant, which pushes you to high frequency, but that makes getting power to it harder. Finding that sweet spot is critical.
What sweet spot did they find for this specific 32-nanometer SOI process they used?
They determined that a resonator frequency of 270 megahertz offered the best trade-offs.
270 megahertz. Okay.
And that corresponds to needing about a 157 nanohenry inductor and a 2.2 picofarad capacitor.
Which still need to fit on that tiny 300 by 300 micron chip.
Precisely.
Yeah.
So to shrink the resonator further, they very cleverly designed it using a 300 by 300 micrometer inductor printed right onto the chip.
The inductor takes up the whole chip area.
Pretty much. And they carefully tuned its parasitic capacitant, the capacitance that just naturally exists between parts of the coil, to contribute to the overall resonance circuit.
So they use the unavoidable parasitic effects to their advantage.
Exactly. It's an integrated approach, essential for hitting that size target while still harvesting power effectively.
Okay, so the tiny coil on the chip receives the 270 megahertz wireless power. But that's AC, right? The chip needs DC. How does it convert it? That's the power harvesting circuit in Figure 2.
Correct. It's a two-stage process shown there. First, circled in red, is the charge pump rectifier.
Like a tiny pump for electrical charge.
Sort of, yeah. It takes those incoming AC waves of energy and converts them into a usable DC voltage. It uses a specific design, a cross-connected bridge rectifier with special transistors.
Special how?
They're called thick oxide low Vth transistors. Low Vth means low threshold voltage.
So they turn on really easily.
Yes, with very little voltage. Vital for starting up and working efficiently at the extremely low power levels involved here. This design allows operation up to 2.1 volts unregulated and makes the internal diodes more efficient.
How efficient is this rectifier stage?
The simulation showed an impressive 80% power conversion efficiency. That's converting the 270 megahertz RF input to a 1.1 volt DC output while drawing 1 milliamp.
80% is really good for something this small operating wirelessly.
It's remarkable, yeah. Shows meticulous low power design.
Okay, so now you have DC voltage, but it's unregulated. Electronics need a super stable voltage, right?
Absolutely. Fluctuations can cause errors. That's the job of the next stage, circled in purple in Figure 2.
Yeah.
The low dropout regulator, or LDO.
The LDO.
Yeah.
Like a voltage governor.
Exactly. It takes the potentially fluctuating DC from the rectifier and provides a perfectly stable, regulated voltage, usually 1 volt, for the chip's core logic and sensors.
Essential for reliable operation.
Totally.
Yeah.
And they added a compensation capacitor in a clever way to improve stability and reduce the need for a big output capacitor.
Again, saving space. Every micrometer counts.
Always.
How much power does this LDO itself consume just doing its regulation job?
Just 25 microwatts for a regulated 1 volt output.
Tiny.
Incredibly low, which helps with overall efficiency and, maybe more importantly, minimizes heat generation. Critical for a brain implant.
Absolutely. So we have the internal power conversion sorted. What about getting the power from outside to the chip, the external transmitter coil?
Right. You need an optimized external transmitter coil placed on the scalp, positioned above the implant.
How efficient is that wireless link?
Well, they measured it. With the external coil placed 4 millimeters above the chip, a realistic distance through scalp and skull,
Yeah.
the power transfer efficiency was measured at 0.002% when delivering 1 milliwatt to the chip.
Ooh, 0.0002%. That sounds incredibly low. How can that possibly be enough power?
It does sound tiny, but remember two things. First, the chip itself sips power just microwatts total.
It's super efficient.
Incredibly efficient. And second, the external transmitter can pump out significantly more than 1 milliwatt.
Ah, so you just transmit more power externally to compensate for the low transfer efficiency.
Exactly. Even that tiny fraction is enough to run the ultra-low power device. It really highlights how crucial that efficient internal power harvesting and regulation is. They make the most of every bit of energy that gets across.
Right. It's a testament to the whole low power design philosophy.
Yeah.
Okay, powered up. Now, the eyes, the pixel array. How does it capture images while still being incredibly low power? Because low power inside the brain isn't just about battery life, it's about safety, right? Heat.
It's absolutely critical. The paper really emphasizes this. Super miniaturized systems like this are terrible at dissipating heat. Tiny surface area embedded in tissue.
Nowhere for the heat to go.
Exactly. So unconstrained power dissipation could locally raise tissue temperature.
Which could damage neurons or affect their activity.
Precisely. Undesirable physiological effects. So minimizing power isn't just efficiency, it's fundamental safety and data integrity. Every microwatt saved is less heat generated.
Makes perfect sense. So how do they achieve such low power, especially with image data? Isn't transmitting images usually power hungry?
Hugely power hungry, and this is where the real genius of their data compression strategy comes in. Pixel-wise coded exposure, or PCE, imaging.
PCE imaging, okay.
In any wireless implant, data transmission is often the biggest power hog. Power use is directly proportional to data rate.
So less data transmitted means less power used.
Dramatically less. If you can slash the data rate, you slash the power needed for transmission.
How does PCE slash the data rate?
It compressively samples the scene right at the pixel level, instead of every pixel recording flat out all the time.
Generating tons of data, most of which might be background noise or no change.
Exactly. PCE implements spatiotemporal compressive sensing. It leverages the fact that neural activity is often sparse.
Sparse meaning only a few neurons are active at any given moment.
Right, or changing significantly. So PCE allows for sub-Nyquist sampling. You capture fewer samples than traditional theory would suggest you need.
But you can still reconstruct the full video accurately.
Yes, because you know the signal is sparse. It's like an intelligent camera focusing only on the changes, the important bits, vastly reducing the raw data generated.
That's fascinating. Only recording what's essential. How do they control which pixels record when? The paper mentioned an extra transistor.
Yes, Figure 3 shows it. Each pixel gets an extra exposure control transistor called EX.
Okay.
When EX is high, the pixel works like normal resets, integrates light. But when EX is low, the photodiode gets disconnected from the readout part.
Ah, so it holds its charge.
Effectively, yes. Its exposure is prolonged until EX goes high again, by precisely timing this EX transistor for each pixel individually.
You control each pixel's exposure duration.
Exactly. You can create complex, coded patterns of exposure across the array.
That's very clever pixel-level control. And what kind of compression did they achieve with this?
Pretty remarkable results. Using pseudorandom exposure patterns, they showed they could compress a 30 frames per second video down to less than 3 FPS equivalent samples.
Wow, that's more than a 10x compression rate.
It is. Greater than 10x. And the impact on power. A 10x reduction in data rate can cut transmission power by 10 decibels.
That is a massive power saving, crucial for an implant.
Absolutely transformative for making it viable.
So how is this PCE array actually built on the chip? Figure 4 shows the implementation.
Right. Figure 4 shows the circuit. Each pixel has a 1-bit memory cell to control its EX transistor.
So you can load patterns into the pixels.
Exactly. There are also standard reset, RST, and row select RSEL signals. The prototype array has 256 by 256 pixels.
That's a decent resolution for this size.
It is. They used SRAM drivers to load the exposure patterns into the pixel memory. For testing, they multiplexed pixel callers to analog output pins.
And how much power does the array itself use, just capturing the image?
The whole 256 by 256 array consumes around 300 microwatts at 1.4 volts. Again, incredibly low for the actual image capture.
Okay, now diving right into the pixel's core, the photodiode. What type is it?
It's that lateral PIN structure we mentioned earlier with SOI, a lightly doped P-type channel between heavily doped N and P regions.
Designed to be thin and capture light traveling sideways-ish.
Right, and the pixel pitch, the center-to-center distance between pixels, is just 4 micrometers. Tiny, allowing high density.
Very high density for good spatial resolution in that small footprint.
Now, the paper compared SOI to SOS silicon on sapphire. Why was that comparison important, especially for backside illumination?
It's a crucial distinction. These lateral diodes in SOI trade some quantum efficiency for thinness. Now, SOS has also been used for similar things, and SOS has a natural advantage. Sapphire is transparent to visible light, so you can do backside illumination on SOS easily, no extra steps needed.
But SOI isn't like that.
No. The SOI structure, while thin at the device layer, sits on a thick silicon handle substrate, and that handle substrate is opaque.
Ah, so even though the active part is thin, the base it sits on blocks light from the back.
Exactly, which is a huge problem if you need backside illumination because you put the coil on the front.
So SOI gives you the thin device layers and lets you put components on top, but inherently blocks the back view.
Correct, which means you can't just use it for backside sensing off the shelf. It requires a major complex post-processing step. Okay, so this brings us to that crucial step. Backside illumination is essential because the coil's on top, but the SOI wafer blocks the back. How did they solve this? How did they get light to the sensors from behind?
You've nailed the problem. Front-side illumination is out. So they developed a specialized, multi-step post-processing flow.
Post-processing meaning after the chip is made by the foundry.
Exactly. They basically have to physically remove that opaque bulk silicon handle substrate, make the back of the chip transparent.
That sounds incredibly delicate, like microsurgery on the chip itself.
It is. Very intricate, very precise, especially in something already so small and thin.
Can you walk us through that process? Figure 5 details it.
Absolutely. It's quite advanced, and they do it on individual dies, not whole wafers, adding complexity.
Okay, step 1.
Step 1, they take the dies from the foundry and mechanically grind or polish them down, thinning the whole thing to about 30 micrometers overall thickness.
Grinding it down carefully.
Very carefully. But mechanical grinding isn't precise enough to stop perfectly at that buried oxide layer, which is only 145 nanometers thick.
Right, it leaves a thin, uneven silicon layer still blocking the view.
Exactly. Still opaque, so that needs to go.
How do they get rid of that last bit of silicon precisely?
That's where controlled wet etching comes in, using a chemical, tetramethylammonium hydroxide, or TMAH. This chemical etches away silicon, and the buried oxide layer acts as a perfect etch stop.
Meaning the etching stops automatically when it hits the oxide.
Largely, yes. It removes the unwanted opaque silicon right down to the transparent oxide layer, exposing it for backside illumination without damaging the active circuitry above it.
Why TMAH specifically? What are its advantages?
Good selectivity, it mainly eats silicon, and crucially, it's CMOS compatible. Unlike other etchants like KOH, it doesn't have mobile ions like potassium that could contaminate and ruin the CMOS transistors.
Ah, compatibility is key.
Yeah.
But were there any challenges using TMAH?
Yes, one big one. TMAH also tends to etch aluminum.
And the contact pads on the chip for testing are aluminum.
Exactly. You need those pads to test if the chip even works before you do all this aggressive thinning. But the thinning chemical would destroy the test points.
That's a catch-22. How do they solve that?
A very clever trick from prior research. They dissolve some silicon powder into the TMAH solution itself.
Silicon powder in the etch stand.
Why? Because it forms aluminosilicates right on the surface of the aluminum pads. This creates a protective passivation layer.
So it coats the aluminum pads, protecting them from the TMAH etchant.
Precisely. This allowed them to test the chips fully before thinning, ensuring they weren't wasting effort on dead chips. Really ingenious microfabrication problem solving.
That is incredibly clever. So after all this grinding and etching, what's the final state? Is the chip protected?
Yes. After they remove the original carrier wafer and dice away the edges with the pads, the remaining tiny chip is essentially encapsulated in silicon dioxide, the buried oxide layer.
And silicon dioxide is pretty tough.
It's quite inert and has low water vapor permeability. This means, remarkably, the finished device can be used for acute experiments, short-term studies, without needing any extra bulky encapsulation coatings.
Wow, so it's naturally sealed enough for initial use. That's a bonus.
Big bonus. Further reduces profile, simplifies use for those first critical tests, a self-protected microsensor.
Fantastic. So they have this incredibly thin, back-illuminated chip, power, data compression. What did the preliminary tests show? Did the photodiodes work? Focus 6 has some results.
Initial tests focused on just validating the photodiodes. They started with front-side illumination on a prototype chip where the light wasn't blocked yet.
Okay.
Figure 6 shows results from scanning a column of 20 pixels. You can clearly see the pixel output change between dark and illuminated conditions.
So the basic light sensing worked.
Yes, it clearly demonstrated the lateral PIN photodiodes were operational. There was some non-uniformity pixels responding slightly differently, but that's common in early prototypes and usually correctable with calibration software.
Okay, so front side worked. What about the crucial backside tests, the whole point of the thinning?
That's where they hit some practical snags, especially with their larger 3mm by 3mm test chip.
Larger. Why use a larger one for testing?
Easier to handle and connect to initially, but when thinned below 30 microns, even that larger chip became incredibly fragile, like handling microscopic glass.
Ah, so connecting wires for testing became difficult.
Exactly. Wire bonding, the process to connect the chip to test equipment, exerted too much force on the delicate, thinned silicon. It caused breakage.
Ouch. So they couldn't easily complete the backside illumination tests on those ultra-thin prototypes.
They faced difficulties, yes. But the paper states they're actively developing gentler handling and bonding methods for these fragile dies.
So work in progress on the final validation step.
Right. They fully expect good pixel response from back illumination, given the design and the front-side results. It just shows they're really pushing the physical limits of manipulating materials at this scale.
Okay, so feasibility shown, innovative design, promising initial results, especially on power and compression, but still a prototype. There must be hurdles before this is ready for routine animal studies, let alone anything clinical.
Absolutely. It's pioneering work. Always more challenges.
What's the biggest remaining one they highlighted?
The paper pinpoints a major one, the fluorescent excitation light source.
Right.
Yeah.
The sensor detects the fluorescence, but something needs to cause the fluorescence in the first place.
Exactly. You need to deliver excitation light into the brain tissue to make those GECI-labeled neurons light up.
So the question becomes, what's the best way to get that light in there efficiently, minimally invasively, and without causing heat issues itself?
That's the critical missing piece for a fully integrated standalone system.
What are some possible solutions being explored for that integrated light source?
Several active areas. One is using tiny light-delivering optical fibers, common in optogenetics.
Okay.
Another exciting path is implantable micro-LEDs, tiny light sources fabricated very close to or even on the sensor chip itself.
An integrated light source.
Yeah, or physically attaching a micro-LED to the recording device, but
each has pros and cons.
definitely. Trade-offs in invasiveness, power needed for the light delivery efficiency through tissue, and again, heat generation from the light source itself. Finding the optimal method is still a very active research area.
A complex puzzle, balancing all those factors. But the core sensor tech itself, the thinness, the wireless efficiency seems incredibly promising.
It absolutely is. The authors really demonstrated the feasibility, the concepts, the preliminary results. It's a foundational step, breaking those previous barriers.
What's the immediate next step for them?
Completing those crucial backside illumination tests to fully validate the thinning and sensing. And then developing a standalone chip that integrates everything, power, PCE pixel array, and eventually an optimized light source into one tiny functional device.
The future is definitely bright and incredibly tiny.
Couldn't have said it better myself.
Okay, so let's recap. We went from the big challenges of watching neurons in action through the limits of old tools to this incredibly elegant miniature solution. A 25-micron thick, wireless, backlit sensor you can inject into the brain. So what does this all mean for you, the listener, the curious mind, trying to understand the brain?
Well, like we said, it's not just a tiny camera. It feels like a potential paradigm shift.
Oh, so
It offers that dream combination, cellular resolution with minimal invasiveness. That's been the holy grail for so long.
and connecting that to the bigger picture.
Imagine the possibilities. Understanding exactly how memories form, watching neurodegenerative diseases like Alzheimer's progress cell by cell in real time.
Or even getting at the mechanisms behind complex behaviors, consciousness.
Exactly. Moving from seeing the orchestra section to seeing the individual musicians playing their unique parts in the brain's symphony.
Making our observation almost invisible, yet more revealing.
That's the goal, yeah. Paradoxically more revealing because it's less disruptive.
It is truly revolutionary to think about. Imagine that future. Seamless, cellular-level brain monitoring, in real time, in living beings, no wires, no bulky gear. What kind of ethical questions does that deep access raise for us?
That's a profound question we'll need to grapple with.
Well, what unforeseen discoveries might leap out when we can observe neural activity with such clarity and over long periods?
Yeah.
How might it change our fundamental understanding of ourselves? Or what new frontiers in brain-computer interfaces, in diagnostics, maybe even therapies, could this kind of unobtrusive tech suddenly unlock? What possibilities, what new questions does this spark in your imagination?